Some of the biggest breakthroughs in science are really just fantastic detective stories. And today, we're going to dive into one of those stories. It's about a brilliant idea that was lost for decades, and the crucial clues that finally showed us how life's instructions are physically passed down. So, think about it. After Gregor Mendel did his incredible work with pea plants, science was left with this massive cliffhanger. He described these invisible factors that pass traits from parents to kids, but what were they, physically? Were they tiny particles? Some kind of fluid? Nobody had a clue. They were just an abstract idea, and the race was on to find out what they were really made of. All right, so here's our game plan for cracking this case. We'll start with Mendel's mysterious idea, then we'll check out the new clues that started popping up under the microscope. We'll get to that big aha moment when everything clicked, which led to the theory itself, and finally, we'll meet the tiny little fly that basically proved it all. Okay, let's rewind. Our story kicks off with the man himself, Gregor Mendel. But his groundbreaking discovery didn't exactly make a splash. Nope. In fact, for a long time, it was completely ignored. So why the 35-year silence? Well, a couple of big reasons. First, you couldn't just tweet your results back then, right? His work just didn't get around to the right people. Second, his idea that traits weren't just a blended soup of mom and dad, but were passed on as separate little packages, that was totally revolutionary. And on top of all that, he used math to explain biology. People just weren't doing that yet. He was simply way, way ahead of his time. So the key thing here is that Mendel proved these heritable units existed through his experiments. He called them factors. But he couldn't point to one under a microscope or say where it lived in the cell. And that was the central mystery that needed to be solved. Where were these things? So decades go by. Mendel's paper is sitting on a shelf somewhere collecting dust. But in the background, technology is taking some huge leaps forward. Microscopes were getting better, and new techniques for staining cells were about to uncover the next big piece of the puzzle. Just look at this timeline. You've got this long, quiet period, and then bam, right around 1900, everything just explodes. First, 3 different scientists independently rediscover Mendel's work. And then, just 2 years later, we could finally see and describe these little structures moving around the inside of cell's nucleus right before it divided. And those structures were called chromosomes. The name is pretty cool. It literally just means colored bodies, because they soaked up the chemical dyes scientists used to see them. And people were just glued to their microscopes, watching these chromosomes do this perfectly choreographed dance, doubling and then splitting every single time a cell divided. It was so orderly. So predictable. You just knew they had to be important. But what exactly was their job? Okay, now this is where it all comes together. This is the big aha moment. You've got these two completely separate ideas floating around. Mendel's abstract factors on one hand, and these very real, visible chromosomes on the other. And they are about to crash into each other in the best way possible. So, in 1902, you have 2 different scientists, Walter Sutton and Theodore Boveri. They're both studying meiosis. That's the special process that makes reproductive cells, like sperm and eggs. And here's the crazy part. Working totally separately, they both had the same incredible realization. The way chromosomes were moving around, it was a perfect mirror image of how Mendel said his factors should behave. I mean, just look at this comparison. It's uncanny. On the left, what we can see chromosomes do. On the right, what Mendel predicted his genes would do. Let's run through it. Chromosomes, they come in pairs. Mendel's factors, they come in pairs. Check. Chromosomes get separated when making sperm and eggs. Yup. Mendel's factors do too. Check. Different pairs of chromosomes sort themselves out independently of each other. You guessed it, so do Mendel's factors. The parallels were just too perfect to be a coincidence. So let's actually watch how this segregation works. This diagram makes it super clear. Over on the left, you've got a parent cell with its pairs of homologous chromosomes. Think of them like pairs of socks, one from each parent. Now, in that first big split, meiosis I, the pairs get separated. One whole chromosome goes one way, its partner goes the other. Then, in meiosis II, the 2 identical copies making up each chromosome get pulled apart. What are you left with? 4 new cells, each with only 1 chromosome from every original pair. This physical process is the perfect explanation for Mendel's law of segregation. And here's the other key principle, independent assortment. This is all about the shuffling. It's basically asking, how do the pairs line up before they split? And the answer is, it's totally random. Check out possibility I. The long orange lines up near the short red, but in possibility 2, there's nothing stopping that long orange one from lining up with the short green one instead. How one hair orients itself has zero influence on the others. It's this random shuffling that is the physical basis for independent assortment, and it's why siblings can get such a different mix of traits from the same parents. So, with all this powerful evidence staring them in the face, Sutton and Boveri were ready to make it official. They were about to propose a huge idea, one that would finally connect the world of cells with the world of genetics. And there it is, the chromosomal theory of inheritance. It's a beautifully simple but profound idea. It says that Mendel's factors, what we now call genes, are physically located on chromosomes, and the way those chromosomes move around during meiosis is the physical reason we see the patterns of inheritance that Mendel discovered. It was a perfect marriage of two different fields of science. But you know, in science, a brilliant theory is just a starting point. You need proof, called hard experimental evidence. And the final piece of this puzzle came from a very unlikely hero, the common fruit fly. Meet Thomas Hunt Morgan and his lab partner, the fruit fly, Drosophila melanogaster. And what's great is that Morgan was actually a skeptic at first. He wasn't totally sold on the theory, which made him the perfect person to put it to the ultimate test. He set out to find variations in fruit flies and trace how they were inherited, really putting the theory through its paces. So why these little flies? Well, they turned out to be the perfect subject for this kind of research. I mean, they're cheap, they're easy to grow, they have a new generation every 2 weeks, and a single pair can have hundreds of babies. This meant Morgan could get mountains of data really fast. Plus, they had lots of easy-to-spot traits, like eye color, that he could track from one generation to the next. So, to sum it all up, Morgan's work with these tiny flies was the definitive proof. We now knew, for sure, that genes have specific addresses or locations on chromosomes. We knew that these chromosomes are the physical vehicles of hereditary. And most importantly, their observable movements during meiosis explained Mendel's abstract laws perfectly. The mystery of the invisible factors was finally solved. But, of course, solving one giant puzzle in science usually just reveals another, even more interesting one. And that leaves us with a real head-scratcher. If genes are physically stuck together on the same chromosome, kind of like beads on a string, how on earth could they possibly follow Mendel's law of independent assortment? Now that sounds like a puzzle for another time. Thanks for joining me.